Hi, Michelle Glass here. Welcome back to another episode in our digestive system video series, chapter 24, lecture 24. Excuse me, this episode is all about the regulation of the digestive tract. So it begins with the cephalic phase. Cephala, remember, is talking about the head. So this is your cerebrum. This is your sensory awareness of food. So seeing food, smelling food, tasting food, thinking of food, all of course directed by the central nervous system, will start your stomach juices flowing. So you're going to increase your gastric secretion. That means hydrochloric acid and pepsin. You're going to release the hormone gastrin, which remember is made by those G cells in the stomach. Gastrin, remember, is increasing gastric secretion and also gastric motility. So you're preparing for that eventual food that's going to come into the stomach. Once you have food coming into the stomach, you enter the gastric phase. So the arrival of food in the stomach is going to signal the stomach to really get busy. This is lasting for about 3 to 4 hours. Remember, one of the jobs of your stomach is to hold that food and then just release small amounts into the small intestine so you have adequate time for good digestion and absorption of nutrients. The arrival of food in the stomach is going to stimulate the gastric phase because of 3 things. So the actual distension of the stomach is going to stretch receptors. The increase of pH from that bolus entering the stomach will signal chemoreceptors, and also the arrival of undigested food material into the stomach will also signal the gastric phase to begin. The gastric phase is coordinated by gastrin. So this is, again, continuation of the increase of the gastric secretions, hydrochloric acid, pepsin, and also motility, the churning, mixing, segmentation movement of the stomach. Okay, the actual distension of the stomach, so the stimulation of those stretch receptors will signal what's called the gastroenteric reflex. This is where you're going to increase the motility and secretion along the entire length of the small intestine. So, of course, as material moves into the stomach, we know we're going to be sending some of that material onto the small intestine. So we need to move it along down that assembly line. You also have a gastroileal reflex, and this is going to open up that ileocecal valve. So as material is moving in the small intestine, it will make its way also moving into the large intestine. So this is how, eating food is going to stimulate movement along the entire length of the GI tract. Finally, as the chyme enters the duodenum, we get into our intestinal phase. This is going to signal our enteroendocrine reflex. So, of course, this is the release of all of those hormones that we've talked about coming from the duodenum: secretin, gastroinhibitory peptide, vasoactive intestinal peptide, secretin, CCK. Did I say secretin twice? Maybe. Enterokinin. So all of those that we've labeled, they're going to inhibit the activities of the stomach. So inhibit gastrin, as well as, then slowing down gastric secretions and motility. You're going to have contraction of the pyloric sphincter. So this is going to prevent additional material from entering the duodenum and also increase the duodenal mucus production, because now you've got this acidic chyme coming into the duodenum, and we really need to raise the pH of that material. During this whole period of time when you are absorbing nutrients from your meal, so about 4 hours after your meal, you're going to be in what's called the absorptive state. So if you're eating a breakfast and a lunch and a dinner, then for about 12 hours every day, your waking hours when your cells are most metabolically active, you would expect to be in the absorptive state. Now, there are several hormones coordinating the absorptive state, but let's think primarily of insulin. Insulin is the hormone that's released by the beta cells of the pancreas. It's going to increase your glucose uptake by your cells. Because you're flooding your bloodstream and your cells with glucose. That's our primary source of ATP production. So let's take it while we've got it. And then you're going to increase your triglyceride production because you're going to store those lipids for times when glucose is not as readily available. And we have plenty of ATP to make lots of protein. So you're going to increase your uptake of amino acids and increase your protein synthesis. Growth hormone and also your sex steroids, either estrogen for females or androgens such as testosterone for men, will increase the amino acid uptake to help with that protein synthesis. So we're seeing that these hormones are kind of companions to the work of insulin. After you are, in bed, so you've been eating and working all day long and in this absorptive state, and then you are resting and you're no longer eating and you've already digested and absorbed all the nutrients from your food, you're going to enter what's called the post-absorptive state. In this case, you are restricted to whatever you have stored in the body, so on your internal reserves. Now that's okay because your metabolism is typically low here if you're resting, and so you have plenty of time to really mobilize your energy reserves. And then in order to maintain your normal blood glucose levels, you're going to do a lot of what are described as glucose-sparing activities, and we've talked about this previously. The primary hormone involved here is glucagon, but also helping glucagon, you have epinephrine, you have glucocorticoids, and also growth hormone is assisting here. The glucagon is doing the opposite of insulin, so it's going to increase the breakdown of your triglycerides so that you can release fatty acids into the bloodstream so that your cells can use the fatty acids to make ATP because glucose is limited. You're going to increase your breakdown of glycogen. This is your polysaccharide in your liver and skeletal muscle so that you're releasing some glucose there. Your liver cells in particular can make new glucose, so they can use amino acids or they can use lipids in order to construct glucose. So you're going to increase your gluconeogenesis. And also you're increasing the breakdown of your proteins because you can take those amino acids and use them to make glucose. Also, you can take the amino acids and use it directly to make ATP. So when we are in the post-absorptive state, we transition to more glucose-sparing activities, saving that glucose for your neurons, the cells that really need it, and pretty much telling the rest of your cells that they can use your stores of fatty acid. As part of this glucose-sparing activity, your liver is breaking down lipids and amino acids as part of that gluconeogenesis process, as well as, lipids and amino acids can be used to make ATP. How our cells use lipids and amino acids to make ATP is to convert them into what are called ketone bodies. So if you've heard of ketosis, it's talking about the formation here of ketone bodies. Okay, that's it. Stay tuned.